Hi, I'm Medina Fadl, and I'm here to present DGrowthR, which is a comprehensive statistical framework for analyzing large-scale microbial growth data. This R package is a collaborative work with the colleagues that are also listed here. So typically in microbiology, we study how bacteria grow under different conditions. We grow strains with various compounds and then measure growth over time. But analyzing these growth curves is quite challenging. They show complex dynamics that simple models cannot capture. Standard approaches, like for example the logistic models, assume rigid shapes. And in this case, they often fail with real-world data. And as an example here, we looked at Vibrio cholera growth curves under various compounds. So on the left, you can see control condition, which mostly looks like a textbook growth curve. We observe a nice sigmoidal shapes, and traditional models would work perfectly fine here. In the middle, the antibiotic amikacin was added, and one can see that we can observe now irregular patterns. The bacteria is growing and then struggles, and most of the models would miss this completely. The situation becomes even more dramatic when we look at penicillin. So we observe an initial growth state and then a peak followed by a cell death phase. And as you can see here, the logistic model is not able to capture this correctly. So what this implies is that real-world complexity demands flexible approaches. Apart from that, we're facing a couple of more challenges. So typically, in a high-throughput experiment, one can generate hundreds of replicates, and these must be analyzed together properly. Third, we would also like to know if the difference between conditions are actually real and statistically valid. And then fourth, we're dealing actually with thousands of growth curves per experiment, so manual analysis in this case is not feasible. So to address all of these challenges, we developed DGrowthR, which consists of 4 integrated components, but do not necessarily all have to be applied. So part A is the data import, which handles any plate reader format and automatically organizes thousands of growth curves. Part B consists of preprocessing and exploration. And the preprocessing steps here are optional, but could include log transformation, baseline removal, or time point adjustments. In the low-dimensional embedding, we offer UMAP and functional principal component analysis in order to reveal patterns in the full dataset. The heart of DGrowthR is actually part C, which is the Gaussian process regression. In this case, the Gaussian process models each curve flexibly without making any assumptions and includes the multiple replicates. After that, after fitting the growth curves, we can extract biological parameters that are of interest. And then part D is the differential testing framework in which the statistical comparisons between conditions can be made. And then we can look at the results in a volcano plot and then determine which chemicals, for example, lead to growth increase or decrease. Let's look at the Gaussian process approach in a little bit more detail. So unlike a parametric model, we don't make any assumptions about the shape. Instead, the Gaussian process learns directly from the data. And this happens the following way. So we specify a prior over functions and then update it with our observations. This gives us 2 key advantages. The mean function adapts to any pattern, and the confidence band shows the uncertainty. From these fit, we can then extract these parameters, like, for example, the lag phase duration or the maximum growth rate. This can help give insights into when does a chemical have what effect on the growth curves. For comparing conditions, we use hypothesis testing. So our null hypothesis in this case is that growth patterns are the same across conditions. And the alternative is that growth varies by condition. Based on that, we can then calculate the base factor, which tells us how much more likely is the data under each hypothesis. These results can then be validated using permutation tests. In this case, the complete growth curve and the labels based on that are shuffled and used to rebuild a new null distribution. Using this approach, we can then see if the differences are real or just occurred by accident. So let me show you now DGrowthR in action. We analyzed the same bacteria as before under antibiotic stress. And the exploratory data analysis revealed something quite interesting. So the UMAP and functional principal component analysis show 4 distinct growth phenotypes in different colors. And in blue, we have the very non-growing curves, and in pink, those that are not affected by the chemical at all. So we observe a regular sigmoidal shape. And then the yellow and the green cluster are kind of those in-between growers. In addition to that, we discovered these differences correlate actually with genetics. So strains with the CBAS antiphage defense show altered antibiotic sensitivity. So in this case, we here have 3 examples. On the left, we observe that wild type grows better than the deleted CBAS strain, actually statistically significantly different. In the case of amoxicillin, we observed that the wild type suffers severe lysis. CBAS maintains, on the other hand, a steady growth. And for amikacin, both struggle initially, but then the wild type strain recovers remarkably while the CBAS strain doesn't. This actually reveals a connection between phage defense and antibiotics resistant. And we would have missed this without a sophisticated analysis. So in order to summarize, DGrowthR provides a complete growth analysis solution in R in which it handles complex dynamics using flexible Gaussian processes and is able to analyze thousands of growth curves automatically. This package is available on GitHub, but also as an app with an extensive documentation. And if this is something you're interested in, you can always reach out to us and discuss the application, but also contribute to this framework. Thank you.